Protein synthesis. DNA provides a blueprint for the cell structure and physiology. This refers to the fact that DNA contains the information necessary for the cell to build one very important type of molecule, a protein. Most structural components of the cell are made up, at least in part, by proteins, and virtually all of the functions that a cell carries out are completed with the help of proteins. One of the most important classes of proteins are enzymes. Enzymes help to speed up necessary biochemical reactions that take place inside of the cell. Some of these critical biochemical reactions include building larger molecules from smaller components, such as occurs during DNA replication or synthesis of microtubules. It also includes the breaking down of larger molecules into smaller components, such as when harvesting chemical energy from nutrient molecules. Whatever the cellular process may be, it is almost sure to involve proteins. Just as the cell's genome describes its full complement of DNA, a cell's proteome is its full complement of proteins. Protein synthesis begins with genes. A gene is a functional segment of DNA that provides the genetic information necessary to build a protein. Each particular gene provides the code necessary to construct a particular protein. Gene expression is the process by which information encoded into a gene is transformed to a final gene product. The interpretation of genes works in the following way. Recall that proteins are polymers, or chains of many amino acid building blocks. The sequence of bases in a gene translates to an amino acid sequence. A triplet is a section of 3 DNA bases in a row that codes for a specific amino acid. The mechanism by which the cell turns DNA code into a protein product is a two-step process. The first step is transcription, and the second is translation. Transcription is the process that goes from DNA to RNA. DNA is housed within the nucleus, and protein synthesis takes place in the cytoplasm. Thus, there must be some sort of intermediate messenger that leaves the nucleus and manages protein synthesis. This intermediate is messenger RNA, or mRNA. mRNA is a single-stranded nucleic acid that carries a copy of the genetic code for a single gene out of the nucleus and into the cytoplasm, where it is then used to produce proteins. There are several different types of RNA, each having different functions within the cell. The structure of RNA is similar to DNA with a few small exceptions. For one thing, unlike DNA, most types of RNA are single-stranded and contain no complementary strand. Second, the ribose sugar in RNA contains an additional oxygen atom as compared to that of DNA. Finally, instead of the base thymine, RNA contains the base uracil. This means that adenine will always pair up with uracil during the protein synthesis process. Gene expression begins with a process called transcription. Transcription is the synthesis of a strand of mRNA that is complementary to the gene of interest. This process is called transcription because the mRNA is like a transcript or copy of the gene's DNA code. Transcription begins in a fashion somewhat like DNA replication. In that region of DNA, a region of DNA unwinds and 2 strands are separated. However, only a small portion of the DNA will be split apart. The triplets within the gene on the section of the DNA molecule are used as the template to transcribe the complementary strand of RNA. A codon is a 3-base sequence of mRNA. Codons directly encode amino acids. Like DNA replication, there are 3 stages to transcription: initiation, elongation, and termination. Initiation. A region at the beginning of the gene, called a promoter, is a particular sequence of nucleotides that triggers the start of transcription. Transcription starts when RNA polymerase unwinds the DNA segment. One strand, referred to as the coding strand, becomes the template with the genes to be coded. The polymerase then aligns the correct nucleic acid with its complementary base on the coding strand of DNA. RNA polymerase is an enzyme that adds new nucleotides to a growing strand of RNA. This process builds a strand of mRNA. Next is termination. At the end of the gene, a sequence of nucleotides called the terminator sequence causes the new RNA to fold up upon itself. This folding causes the RNA to separate from the gene and from RNA polymerase, thus ending transcription. Before the mRNA molecule leaves the nucleus and proceeds to protein synthesis, it is modified in a number of ways. For this reason, it is often called a pre-mRNA at this stage. For example, your DNA, and thus complementary mRNA, contains long regions of so-called non-coding regions that do not code for amino acids. Their function is still a mystery, but the process called splicing removes these non-coding regions from the pre-mRNA transcript. A spliceosome is a structure composed of various proteins and other molecules. The spliceosome attaches to the mRNA and splices or cuts out these non-coding regions. The removed segment of the transcript is called an intron. The remaining exons are then pasted together. An exon is a segment of RNA that remains after the splicing process. Interestingly, some introns that are removed from the mRNA are not always non-coding. When different coding regions of mRNA are spliced out, different variations of a protein will eventually result, with differences in both structure and function. This process results in a much larger variety of possible proteins and protein functions. When the mRNA transcript is ready, it travels out of the nucleus and into the cytoplasm. Translation is from RNA to protein. The codons on a strand of mRNA must be translated into the amino acid alphabet of proteins. Translation is the process of synthesizing a chain of amino acids called a polypeptide. Translation requires two major aids. First, a translator, the molecule that will conduct the translation, and second, a substrate on which the mRNA strand is translated into a new protein. Both of these requirements are fulfilled by other types of RNA. The substrate on which translation takes place is the ribosome. Remember that many of the cell's ribosomes are found associated with the rough endoplasmic reticulum and can carry out the synthesis of proteins destined for the Golgi apparatus. Ribosomal RNA, or rRNA, is a type of RNA that, together with proteins, composes the structure of the ribosome. Ribosomes exist in the cytoplasm as two distinct components, a small subunit and a large subunit. When an mRNA molecule is ready to be translated, the two subunits come together and attach to the mRNA. The other major requirement for protein synthesis are the transfer RNAs, or tRNAs. Transfer RNA is a type of RNA that ferries the appropriate corresponding amino acids to the ribosome and attaches each new amino acid to the last one, building the polypeptide chain, one amino acid at a time. Thus, tRNA transfers specific amino acids from the cytoplasm to a growing polypeptide. The tRNA molecules must be able to recognize the codons on the mRNA and then match them with the correct amino acid. The tRNA is modified for this function. On one end of its structure is a binding site for a specific amino acid. On the other end is a base sequence that matches the codon specifying its particular amino acid. This sequence of 3 bases on the tRNA molecule is called an anticodon. For example, a tRNA responsible for shuttling the amino acid glycine contains a binding site for glycine on one end. On the other end, it contains an anticodon that complements the glycine codon. GGA is the codon for glycine, and so the tRNA's anticodon would read CCU. Equipped with this particular cargo and matching anticodon, the tRNA molecule can read its recognized mRNA codon and bring the corresponding amino acid to the growing chain. Much like the process of DNA replication and transcription, translation consists of 3 main stages: initiation, elongation, and termination. Initiation takes place with the binding of a ribosome to the mRNA transcript. The elongation stage involves the recognition of tRNA anticodons with the next mRNA codon in the sequence. Once the anticodon and codon sequences are bound together, the tRNA presents its amino acid cargo and the growing polypeptide strand is attached to this next amino acid. This attachment takes place with the assistance of various enzymes and requires energy. The tRNA molecule then releases the mRNA strand. The mRNA strand then shifts one codon over in the ribosome. Then, the next appropriate tRNA arrives with its matching anticodon. This process continues until the final codon on the mRNA is reached, which provides a stop message that signals the termination of translation and triggers the release of the new complete synthesized protein. Thus, a gene within the DNA molecule is transcribed into mRNA, which is then translated into a final protein product. Thank you for watching.